Hello, and thank you for joining. In this talk, I will present you a framework that can estimate single cell trees alongside cell state transition dynamics from lineage tracing and RNA-seq data. I will first provide a bit of context and motivate the framework by highlighting some of the challenges that we want to address. Then I present the framework itself and some simulation-based validations. Finally, I will share some preliminary work in cerebral organoids. In the biological reality, on the way from the zygote to a fully grown organism, there are three main cellular event types. These are cell division events, indicated here as bifurcations, cell type change events, here shown as color change events, and cell death events, here shown as their branches. Now, in the last couple of years, several experimental studies have been able to combine single-cell lineage tracing with single-cell RNA-seq, providing us with partial information on these events. But now, how can we best analyze this combined data? There are several challenges involved when analyzing this data, and I'll show you here those challenges that our method can address. One challenge is barcode homoplasy. So that means a barcode or a scarred barcode arising multiple times independently, even though the cells do not share any ancestral relationship. And we address this by modeling barcode evolution, and this allows us to weigh different barcoding or scarring outcomes. Another challenge is that we usually sequence only a subset of the cells. In our method, we have a parameter, the sampling proportion, where we can directly specify how many cells of the whole cell population we have sequenced. Another potential challenge is clone size heterogeneity. However, we model the population dynamics by using a multi-type birth death model. And under this model, the population size or the clone size is stochastic. So the three challenges I've just presented, I borrowed from Shou and Wang's preprint, and I'd like to add two more. Another challenge is that the distribution of cell types at a particular time point is not only the outcome of the cell type change dynamic, it's also the outcome of the cell division and death dynamics. And so by using this multi-type birth death model, we can account for this. Finally, when we reconstruct a lineage tree from lineage tracing data, there are usually multiple trees that can explain the data well. And so in our Bayesian approach, we can account for this uncertainty. Let's have a look at this method in a bit more detail. So to perform Bayesian phylogenetic inference, one needs the following input: a set of cell states and an alignment of the barcodes for all these cells. And then we need the following model parameters: a tree, a scarring model that captures how an unedited barcode can transition into different scarred states, and a population dynamic model. In our case, we use the structured birth death model, which is parameterized by cell division rates for a particular cell type, death rates, and cell type change rates. The posterior distribution is now our quantity of interest. And this is nothing but the probability of our model parameters given our input. And this posterior factorizes into the tree likelihood and the phylodynamic likelihood and some parameter priors. And now the tree likelihood is the probability that we see our alignment given a particular tree and a particular set of scarring parameters. And the phylodynamic likelihood is the probability that we see a particular type change along this tree given the set of the population dynamic parameters. This inference approach is used widely in epidemiology and in macroevolution. However, we cannot just import it into the developmental biology setting for the following reason. We cannot assume a model of nucleotide substitution because this is fundamentally different to the CRISPR-induced scarring process on which the lineage tracing methods are based. Therefore, a first step to tackle this problem in a phylodynamic approach is to come up with a model of barcode change. For each barcode, we model the barcode change process as a continuous time Markov process on the state space of unedited barcodes, silenced barcodes, and scarred barcodes. And there could be more different scarred states corresponding to different scar types. Now, we always start with a cell that has an unedited barcode. And then as time goes by, barcodes can either be scarred during a particular scarring window that is usually given by the experimental conditions. And barcodes can also be silenced throughout the whole time point. Something that's really nice is that we can analytically calculate the transition probability matrix for such a model formulation. And so this makes also the computation of our tree likelihood relatively fast. We validate our model using well-calibrated simulations. That means we simulate trees under this homogeneous population model, and then we simulate alignments along the trees, and then we perform inference and hope that the parameters match the truth. I plot here the results of the simulation study, and on the x-axis, you see the true rate, and on the y-axis, the estimated rate. And every dot represents the median value of the posterior that we infer for one particular alignment, and the gray bars represent the highest posterior density region. And so ideally, we want these black dots to follow the green line, and the gray bars to always contain the truth, the green represented here by the green line. And we can see that this is the case. So our scarring model parameters are well recovered. And we can look at similar summary statistics about the tree. So for example, the tree height and the tree length. And here the true values are represented by these green dots. And we can see that the posterior intervals almost always contain the true values. And then that especially for the tree length, we really capture a wide range of tree lengths. Now, in the final part, I'd like to show you our initial steps to bring this theoretical framework into the application. And we do this in collaboration with Barbara Treutline's group, with Ashley and Suzanne. And the experimental question we are studying here is neuron generation and brain organoids. This picture is taken from a study from Canton et al. In Nature 2019. And we can see here that cells always start in a pluripotent state, and then they transition through a neuroectoderm, neuroepithelial stage, over a neuroprogenitor stage into the finally differentiated neuron stage. Bringing this data into our phylogenetic thinking, we model the following 4 discrete cell type compartments: a pluripotent compartment, a neuroepithelium compartment, an intermediate progenitor compartment, and a neuro compartment. So they start out growing the organoid from 2,000 induced pluripotent stem cells with at least 1 integration. And this is the vector they use. And so you can see that this is a GFP gene that is targeted by the CRISPR-Cas9 protein. And we can distinguish different GFP genes by this barcoded region. And so we will focus on one particular data set, one particular organoid, which was scarred at day 4 and sequenced at day 32. Right. And so for the more complex case that we are all interested in, I unfortunately have to say that the results are still in progress. Running MCMC on these multi-type models takes a while, but I'm quite hopeful that I will have to present something once the conference takes place. So please ask me in a live Q&A or stop by my poster. Conclusions: we have adapted Bayesian phylogenetics inference methods such that they can be used for developmental biology applications. We have validated this approach in silico. Results in vivo are pending, and we also want to publish this as a package in the BEAST2 framework. This leaves me to acknowledge all the people that have made this work possible. And thank you all for listening. which is an open source framework that provides a lot of these tree sampling mechanistics that we also make heavy use of here. And this leaves me to acknowledge all the people that have made this work possible. This is first and foremost, this is my supervisor, Professor Tanja Stadler, and also Timothy Vogan, who have provided tremendous input on all steps of that I've presented here. Then my collaborators from the Treutlein group, this is Ashley Maynard, also Tobias Gerber when he was here, Susan, and Barbara Treutlein. My collaborators within the group, Antoine Swans and Marc Monceau, figures that I'm very happy to have used BioRender, all of the CEVO group, and you. Thank you all for listening.